Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Leah Rosen, the online editor for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the question and answer window on your screen. After the presentation, we will begin the question and answer portion, and I will ask our speaker your questions. Your questions in the question and answer window will only be visible to myself and our speaker. So thank you for joining us today. It is now my pleasure to introduce our speaker, Jenny England from Thermo Fisher Scientific.
Thank you, Leah, for the introduction. Today, I'm going to be talking about the optimization of an anion exchange method for the large-scale purification of plasmid DNA. Here is a brief overview of the agenda. We're going to be discussing some background, which is an overview of applications and also some of our Thermo Fisher products, the porous AEX resins. We'll go into a AEX design of experiments and followed by some results from dynamic binding capacity with our AEX resins. And once we had points from 2 and 3, we then moved into scaled up verification of the results from there. And then I'll discuss conclusions and future work. So jumping directly into the plasmid DNA overview, the rise of cell and gene therapy has really highlighted the need for a robust purification for plasmid DNA. And it typically is very large in size. So plasmid DNA has very large hydrodynamic radius compared to proteins of similar size. And they range in size from 5 to 10 kilobase pairs, with trends moving towards larger plasmid DNA sizes, so greater than 20 kilobase pairs. In addition, the plasmid DNA also has a large negative charge density, and it is also sensitive to degradation by shear nucleases. Plasmid DNA exists in 3 isoforms, so that's what's shown on the bottom right figure. There is the nicked conformation, the linear, as well as the supercoiled, and the most relevant one is supercoiled, in which there is a vaccine requirement of greater than 80% supercoiled DNA. These vaccine requirements result in unique challenges for plasmid purification. The first one being that the product and contaminants such as genomic DNA, endotoxin, RNA, plasmid isoforms, they're all similar in charge and size, so they're difficult to purify. Additionally, as I mentioned, they're susceptible to shear from, and as well as they have high viscosity limits, and so this also limits operational flow rate. Additionally, plasmids only represent less than 1% of the dry cell mass, and because of this, it's difficult to have these high binding capacities that we're used to in other traditional processes, and so there's typically low binding capacities observed for plasmid DNA with conventional chromatography resins. So jumping into the typical downstream process, as I mentioned, this talk is going to focus on the purification chromatography section, and there is an anion exchange chromatography step. And this is typically ideal for binding polynomic DNA and is used for capture, intermediate, and polishing steps. Additionally, after the AEX, there is a hydrophobic interaction chromatography step, which is used for the isolation of supercoiled DNA. But for the work that I'll be presenting today, I'll be focused on the optimization of the AEX step for the capture of a 5.3 kilobase per plasmid. And so Thermo offers a wide variety of AEX resins. There are the porous resins, which have unique surface chemistries, and they could be utilized, for plasmid DNA purification. And starting from the top, we have our D50 and our PI resins, which are weak AEX resins. These contain primary, secondary, and tertiary amines that are ionizable over a narrow pH range. Our H2 resin, which is the third one on the table, has both weak and strong AEX capabilities in that it contains 60% quaternized polyethelenamine and as well as 40% primary, secondary, and tertiary amines. So it has weak and strong AEX capabilities. And lastly, our HQ resin is a more traditional strong AX resin, and this contains 100% fully quaternized amines. And along with the unique surface chemistries that I mentioned, our resins also contain different pore sizes and base speeds, which we'll discuss in the results as to why we think this can contribute to the different binding capacities that we observe from the study today. And these base speeds are related to the surface area, in which a larger number indicates a greater surface area. So for example, the R550 bead, which is on the XQ resin, will have a greater accessible surface area than the R150 bead. So using our understanding of our AX resins and in collaboration with the Fraunhofer Institute, we executed a AX design of experiments focused on the porous D50, HQ, and XQ resins on a 200 microliter robocolumn scale. And this DOE was looking at loading buffer pH, so we looked at 6 and 7, load conductivity of 40, 50, and 60 millisiemens per centimeter, and also looked at different sample loading densities of 1, 3, and 5 milligrams of plasmid per ml of resin. Jumping into the first set of results, this slide here shows the results for the DOE study of the HQ resin, and we looked at the two different pHs, so pH 6 is shown on the left and 7 is shown on the right, and what we found was that lower recoveries were observed at pH 6, in which we observed less than 100% of recoveries, but we could increase the recovery as a function of load conductivity and also decreasing the load density, in which the maximum recovery was obtained at 60 millisiemens per centimeter and 1 mg per ml low density. On the other side, we have pH 7. We could actually obtain 100% recovery at this pH with a high load conductivity again of 60 millisiemens per centimeter and increasing the load density to 5 mg per ml. This slide shows the results for the porous XQ at those same pHs of 6 and 7. And right away, we can see at pH 6, there's large variability in the recoveries, with the lowest recovery observed at 5 mg per ml sample load density. And this recovery can be improved by decreasing the load density to 1 mg per ml at this pH of 6. We could then achieve a more consistent recovery at pH 7 for the XQ resin across the observed DOE conditions, with the best recoveries obtained at 1 mg per ml load density and 60 millisiemens per centimeter load conductivity. And lastly, we have the results for the D50 resin. This resin showed, again, the best performance at pH 7, where we obtained high recoveries at 60 millisiemens per centimeter, and this was independent of sample load density. And in contrast, we have a pH 6 result as well, in which we saw a strong effect of the recovery on the D50 resin as a function of load density, in which a lower load density achieved greater recovery. So in summary, that's what's shown on slide 11. And so I'm showing the results for the D50, HQ50, and the XQ. And so just across the board, we see that pH 7 was the most ideal loading pH, and a higher load conductivity of 60 millisiemens per centimeter resulted in the optimal recoveries for all three AEX resins. The porous HQ favored a lower sample loading density of 1 mg per ml, while the D50 and the HQ favored a 5 mg per ml sample loading density. They all obtained similar maximum recoveries, but they had slightly different purities of supercoated DNA, in which it was better for the D50 and HQ resin at approximately 73 and 75% purity. So although the DOE provided insight into these ideal loading conditions for optimal recovery, we still wanted to determine the dynamic binding capacities of these resins. So this table, it summarizes the results of our DBC study that we did. Using the same conditions identified from the AUX DOE, we determined them for all the AUX resins on a 1 ml column. We found that the greatest DBC was observed for D50 at about 15.6 mg per ml, followed by XQ at 9 mg per ml and HQ at 2.8 mg per ml. And this is really interesting to us because, as I mentioned previously, there is a difference in the base speed as well as the pore size of each of the resins. And so as you increase the number of the base speed, so 150 to 550, there should be an increase in the surface area. And actually what we found was D50 had the highest binding capacity. So what this data suggests is that it's in fact, a combination of the specific ligand attached to the resin as well as the accessible surface area available for the plasmid to bind, and which is what we think is attributing to the greatest binding capacity of the D50 resin. So based on these results, we took the D50 resin and we selected it for scaled up verification and further optimization. This scaled up verification was done on a 5 ml column at pH 7 with a 10 mg per ml loading density. So the left shows the chromatography results for the trace, and the bottom underneath that trace shows the gels of the corresponding fractions. And these results confirm that the binding capacity of greater than 10 mg per ml, and so this agrees with what was observed in the DOE. We believe that the slightly lower binding capacity of 10 versus 15 could be due to this loss of pDNA in the CIP fraction as shown in the gel and the small peak observed at about 250 mL. And so we took this and said, what can we do next to optimize the recovery of this process and in order to maximize the binding capacity of the D50 resin. So this slide shows the results of a small study that we did to optimize the recovery from the D50 resin. Some initial work has been done in which we've increased the pH of the elution buffer. So originally we did this at 7.0 to 9.0, and also we added EDTA into the elution buffer as well. And we found this improved the recovery to approximately 92%. As well as we did not observe a peak in the CIP fraction. So this was really promising for us to see. In conclusion, the DOE results pointed to an optimal purity and recovery conditions for our 3 AUX resins, the D50, HQ, and XQ. We observed a high binding capacity for all 3 resins, with the D50 having the greatest binding capacity of 15 mg per ml. The initial scaled up verification confirmed this high binding capacity for porous D50, and elution optimization with the addition of 2 millimolar EDTA and increase of pH to 9 helped us to improve the recovery. So these results can guide us to further process and resin optimization for pDNA purification. So future work would be to further optimize this capture step with D50, as well as look into the separation of pDNA isoforms and with our porotic resins. We've done some preliminary work that suggests that the benzyl and the benzyl ultra could be good potential candidates, and also looking into the assessment of these for the isoform separations, and also evaluation of larger size plasmid DNA constructs. So with that, I'd like to thank the Fraunhofer Institute for the collaboration with this work, Johannes Buell and Matthias Nolder, as well as Alejandro Becerra from Thermo Fisher Scientific, Gregor Riker, Oriana Cherova, and John Lee. And with that, I'll take any questions.
Great, thanks, Jenny. So the first question we have is, what method was used to quantify the supercoiled and open circular plasmid DNA?
Right, great question. So we use DNA agarose shells to determine the percentage of supercoiled and open circular plasmid DNA present in all of the elution fractions.
Okay. The next question is, can the porous anion exchange resins be used to separate supercoiled and open circular plasmid DNA?
So we actually have not tested the porous anion exchange resins for separation of supercoiled versus open circular DNA. Based off the initial results that I showed, we think there could be some potential, but that would require some further optimization of experiments. But I think too, we could also investigate looking into the Hick resins as well, as I mentioned.
And what method would you suggest for separation of supercoiled and open circular plasmid DNA?
Right. So as I stated in the previous answer, we think that using our hydrophobic interaction chromatography resins would be a great potential method for separation of these different isoforms. And we've done some preliminary work to suggest that our Hick benzyl and benzyl ultra resins would be good candidates for the separation.
Okay. Well, it looks like we're running close on questions. If you have more questions, go ahead and please type those in. We'll be sending those to Jenny, and she can follow up with you directly. The last question for the webcast is, what size plasmids have been purified by the D50 resin, and would this resin be suitable for purification of larger plasmids, such as larger than 20 kbp?
So right now, as I mentioned, we've only looked at this 5.3 kilobase pair plasmid with our current collaboration with Fraunhofer. However, we're looking to add additional work to purifying larger plasmid constructs, such as 20 kilobase pairs and larger.
Okay. Well, thank you, Jenny, and thanks to our audience for joining us. The recorded version of this webcast will be available for on-demand viewing on our website, and as a registered attendee, you'll receive a follow-up email providing us a direct link. We look forward to having you join us at future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.